Hello students, welcome to Bachelor of Science second year, subject Botany, semester 4, course code BOC 104, course title Plant Physiology. The title of this unit, Unit 6, is Respiration and the module name is TCA Cycle. I am Miss Marina Janice Rodriguez, Assistant Professor from DCT's Dempe College of Arts and Science, Miramar, Goa. In this module, you will be introduced to TCA cycle. You will see the cyclic process of TCA cycle and also the outcome. By the end of this module, you will be able to state the different terms of TCA cycle, outline the process of the cycle and also list the products. TCA cycle, known as tricarboxylic acid cycle, which is also called as Krebs cycle or citric acid cycle, was named in honor of the English biochemist Hans A. Krebs, who proposed that this cycle of reactions was observed, and he said that it was seen in the breakdown of pyruvate in the breast muscle of pigeons way back in 1937. Hans Krebs was awarded the Nobel Prize for this discovery in physiology and medicine in 1953. The process? We all know glycolysis occurs in the cytosol and produces pyruvate. The pyruvate then proceeds to the next step, that is the TCA cycle. But the TCA cycle occurs in the mitochondrial matrix. Therefore, the pyruvate which has been generated in the cytosol has to be transported to the mitochondrial matrix through the impermeable inner mitochondrial membrane, and this is done by a specific transfer protein. Once inside the mitochondrial matrix, pyruvate is decarboxylated in an oxidation reaction by the enzyme pyruvate dehydrogenase. Pyruvate dehydrogenase is a complex molecule. It is made up of three components. 1. Pyruvate dehydrogenase itself, dihydrolipoyl dehydrogenase, and dihydrolipoyl transacetylase. The initial step leading to TCA cycle involves the oxidation and production of NADH2 and carbon dioxide from pyruvate, and the combination of the remaining 2-carbon acetate unit with a sulfur-containing compound called as coenzyme A forms acetyl-CoA. It is this compound, acetyl-CoA, which enters the TCA cycle. This slide shows you the overview of the TCA cycle. In the upcoming slides, we will look at this cycle step by step. The first step, here the 2-carbon compound, that is acetyl-CoA, reacts with a 4-carbon molecule, that is oxaloacetic acid, forming it a 6-carbon compound called citrate in the presence of the enzyme citrate synthase. Citrate is then converted into isocitrate by the action of enzyme aconitase. This conversion takes place in 2 steps. First step involves dehydration, wherein citrate is converted into cis-aconitate, and then again rehydration takes place, wherein cis-aconitate is converted to form isocitrate. Isocitrate is oxidatively decarboxylated in the presence of enzyme isocitrate dehydrogenase to form alpha-ketoglutarate. Here, NAD+ is reduced to form NADH, and the second molecule of carbon dioxide is released. Alpha-ketoglutarate undergoes oxidative decarboxylation to form a 4-carbon succinyl-CoA in the presence of enzyme alpha-ketoglutarate dehydrogenase. During this reaction, 1 molecule of coenzyme A is used up and 1 molecule of carbon dioxide is released. In addition to that, NAD+ is reduced to NADH. In the 5th step, we have succinyl-CoA, which is converted to succinate under the catalytic influence of the enzyme succinyl-CoA synthetase. In the presence of inorganic phosphate, the thioester bond of succinyl-CoA is hydrolyzed. Coenzyme A and energy is released that is sufficient for the synthesis of 1-ATP or GTP. Succinate is further oxidized to form fumarate in the presence of enzyme succinate dehydrogenase. Coenzyme FAD is reduced to form FADH2. Fumarate now reacts with one molecule of water in the presence of enzyme fumarase to form the product malate. And in the final step of the TCA cycle, malate is oxidized back to oxaloacetate in the presence of the enzyme malate dehydrogenase. NAD+ is reduced to NADH2 in this step. Please observe that oxaloacetate will then continue the cycle again when it reacts with acetyl-CoA in the second round. This keeps the cycle going on. Hence, the cyclic nature of this particular process. At the end of TCA cycle, we get different products. Let us have a look at that. The outcome of TCA cycle. Let us remember that it was one molecule of pyruvic acid which had entered the TCA cycle. One molecule of pyruvic acid is made up of 3 carbon molecules. Hence, we get 1 carbon dioxide molecule during oxidative decarboxylation and 2 carbon dioxide molecules during the Krebs cycle or TCA cycle. So this makes up for the 3 carbon molecules which had entered the TCA cycle in the form of pyruvic acid. But 2 molecules of pyruvic acid form 1 molecule of glucose. That means it makes a total of 6 carbon molecules. And hence, a total of 6 carbon dioxide molecules will be evolved from 1 glucose molecule. Looking at the other byproducts, we get NADH2 molecules and 1 FADH2 molecules. These products from the Krebs cycle enter the electron transport chain wherein they are oxidized in the presence of oxygen to produce energy, that is ATP molecules. We come to the end of the TCA cycle. These are my references. Thank you.